Hello guys, welcome back to my lesson. So today we're going to talk about a topic of DNA code of life. So this topic revolves around the process of protein synthesis. So this process is a process where a protein is made, and it means that throughout this topic we will be talking about how your body makes protein. So protein is part of every cell in the body, and it helps the body to build and repair cells and tissues. Protein is a major component of the skin, muscle, bone, organs, hair, and nails. For an example, if your hair and nails are cut off, for them to grow back, you're going to need a protein. Your body is going to make a protein specifically for hair and nails. So let's look at all the biological terms in this topic of DNA code of life. You need to know these biological terms by heart. So we're going to look at the things that are involved in the process of making a protein now. The first thing is a nucleotide, which is a building block of nucleic acids. A nucleotide is a building block of nucleic acids. Nucleic acids are DNA and RNA. So they are made of multiple nucleotides. And if you look at the structure of DNA and RNA, you will see multiple nucleotides combined together. That's why it is called a building block of nucleic acids. So let's label this nucleotide phosphate. We have phosphate on the left-hand side. We have the sugar and the nitrogenous base, because they might ask you in the question paper that you should label it. Now let's look at the structure of DNA and RNA. We have DNA there. It's double-stranded. The natural shape of a DNA is double helix. And the sugar there on the nitrogenous bases is the deoxyribose sugar. And then if you look at DNA, we have nitrogenous bases pairing. So the bond between the nitrogenous bases is weak hydrogen bond. So let's look at RNA here. It is single-stranded, no nitrogenous bases pairing. There's ribose sugar. The sugar there is ribose. And DNA, if they ask you in your question paper who discovered the structure of DNA, you can write Rosalind Franklin, James Watson, or Francis Crick. And if they ask you who took a DNA picture, just write Rosalind Franklin only. So let's talk about the locations of DNA, where you can find a DNA. So you can find a DNA in the nucleus of a cell, and the mitochondrion, and the chloroplast in a plant cell. They will ask you that in a question paper. So now I want us to look at the differences between RNA and DNA. We talked about them. RNA is single-stranded, DNA is double-stranded. The sugar in RNA is ribose sugar, in DNA is deoxyribose sugar. Now remember we talked about the nitrogenous base. Nitrogenous base, there are 5 of them. It's cytosine, guanine, adenine, and thymine. So in RNA, we have cytosine, guanine, adenine, and uracil instead of thymine. So they might give you the structure of DNA like this with nucleotides, and they might write the nitrogenous bases on the left, C, A, T, G, A, and ask you to write the ones on the right. You must know that cytosine, C for cytosine, pairs with G for guanine. If they wrote A, they want the one on the other side. No, it's thymine, because adenine pairs with thymine only in DNA. So in RNA, I will show you how uracil pairs with adenine, because it pairs with it during the process of protein synthesis. Don't forget that we're talking about how your body makes a protein. So now let's look at the structures of DNA and RNA again. They might draw them like this in your question paper. Just know that it's the RNA and it's the DNA. They are nitrogenous bases again. As you can see, RNA is single-stranded, DNA is double-stranded. So let's continue in part 2. This is going to be a part 1. I'm going to make a part 2. Thank you so much for listening. So in part 2, we're going to talk about the process of protein synthesis.